In today's video, we're going to be taking a look at nerve impulses and membrane potentials for A-level biology. Now, the first thing that we need to understand is, what does the term potential mean? It simply refers to the difference in electrical charge across a membrane. So in the resting potential, we have a value of negative 65 millivolts. So that means that the tissue fluid outside the axon is 65 millivolts more positive than the exoplasm. So the exoplasm is 65 millivolts less, has an electrical charge 65 millivolts less than that of the tissue fluid outside the axon. So now let's actually take a look at how we can establish, or how the resting potential is actually established. We'll use the hypothetical situation where in the inside of the axon, so in the exoplasm, right, the cytoplasm of the axon, we have the same number of potassium and sodium ions as in the tissue fluid outside of the axon. So what's going to be happening in order to establish the resting potential is the following. Sodium-potassium pumps will actively transport 3 sodium ions out of the axon into the tissue fluid outside the axon for every 2 potassium ions that come in. So 3 sodium ions going out, 2 potassium ions coming in. Now this is done actively, so using ATP. So we can already see that over a period of time, there will be more sodium ions outside than inside, and there will be more potassium ions inside than outside. So that means that there is going to be an electrochemical gradient being established. Sodium ions will want to come into the exoplasm down the concentration gradient. Potassium ions will want to leave the exoplasm and go into the tissue fluid outside the axon. Now we have these leak channels, okay? There are potassium leak channels and sodium leak channels. However, there are many more potassium leak channels than there are sodium leak channels. So much so that the membrane is actually 100 times more permeable to potassium than it is to sodium. So what's going to happen is that these potassium ions that are now in the exoplasm will diffuse down their electrochemical gradient into the tissue fluid outside the axon. So again, this, along with the sodium-potassium pump, the action of the sodium-potassium pump, and these potassium leak channels, we're building up the positivity outside the tissue fluid, outside the axon, and that tissue fluid outside the axon, compared to the exoplasm. Now, as we mentioned, some sodium ions will also be coming back into the exoplasm down their electrochemical gradient. However, as we mentioned before, there is much less diffusion of sodium into the exoplasm compared to potassium, the diffusion of potassium out of the exoplasm. So these are all contributing to a buildup of a overall positive charge outside the axon compared to inside the axon. Now, in the exoplasm, what is also present are proteins that are negatively charged, and they're called anions. Now, these proteins are too large to diffuse through the plasma membrane. So that's very important to understand, as we will get into it a little bit later on. So positively charged ions like potassium and some sodium ions, because we're probably going to have more potassium ions in the exoplasm. Well, we will have more potassium ions in the exoplasm compared to sodium ions in the exoplasm. So that means that there will more likely be potassium ions being attached to these anions compared to sodium ions. There will also be some sodium ions attached to the anions. However, there will be more potassium ions. So what this is going to do is that these anions will pull, right, because opposites attract, these anions will pull the potassium ions towards them. Now, the potassium ions will be getting pulled out of the tissue fluid, right, to go down their electrochemical gradient. But the pull of the anions will actually be equal to that of the pull of the electrochemical gradient that the potassium ions have, and so the potassium ions will actually be retained inside of the exoplasm, okay, and we will reach an equilibrium. Now, although the sodium-potassium pump is constantly working throughout all of the different potentials, the rate of diffusion of sodium ions back into the exoplasm compared to the diffusion of potassium ions out of the exoplasm will be balanced, so there will be no change in charge, and this is how we reach that equilibrium, the resting potential, and the steady membrane potential difference of negative 65 millivolts. Okay, so these anions are keeping the potassium ions in the exoplasm, and this, along with this will contribute to there being no net movement of ions into versus going out of the exoplasm, which will ensure that we maintain a steady membrane potential difference of negative 65 millivolts, right? The resting potential. Now, it's important to understand that the resting potential can be any value between negative 50 millivolts and negative 90 millivolts. However, it is generally around negative 65 millivolts. Now, what's then going to happen? So we have the resting potential established. What's then going to happen is that there is going to be some form of an electrical impulse that will come as a result of a stimulus. Okay, for example, in that example that we were taking a look at with the chemoreceptors of taste buds, those chemoreceptors will pass on an impulse to the sensory neuron. So what's going to happen is that electrical impulse will cause some voltage-gated sodium ion channels to open. Okay, so that change in electrical charge will cause these voltage-gated sodium ion channels to open. Now, they were initially closed, as were the voltage-gated potassium ion channels. So all of these voltage-gated channels were closed. Now, because we've had that electrical charge, that electrical impulse, the voltage-gated sodium channels open, allowing some sodium ions to enter. Okay, so only a few of the voltage-gated sodium channels will open, depending on the power of that electrical impulse that is coming to this area of the axon membrane. So sodium ions will then diffuse through the voltage-gated sodium channels down their electrochemical gradient, and depending on how many voltage-gated sodium channels were opened, right, this will affect the number of sodium ions that enter into the exoplasm. Now, if sufficient sodium ions diffuse into the exoplasm, and the exoplasm becomes more positive, right, because we're getting positive ions coming back into the exoplasm, we may be able to reach a threshold value, which is at about negative 50 millivolts. So it's 5 to 15 millivolts more positive than the resting potential, so greater than the resting potential of negative 65 millivolts. Now, as those sodium ions will diffuse in, they will cause more voltage-gated sodium ion channels to open, and this is what may enable the threshold value to be reached. Now, this change of membrane potential difference, so it's becoming more positive, is known as the generator potential, and this will occur in the sensory neurons. In motor neurons, the initial change of membrane potential difference up to the threshold value will be called excitatory postsynaptic potential. So we have this increase up to the threshold value. If we reach the threshold value, then an action potential will be stimulated, will be caused. So that will cause all of the many voltage-gated sodium ion channels to open. So the sodium ions will diffuse rapidly, right? They will all just flow into the exoplasm down their electrochemical gradient up to the point of around about plus 40 millivolts. where the membrane is now said to be depolarized. Okay, so initially it was polarized. This was the resting state of the membrane, of the exon membrane at negative 65 millivolts. Now, because we have changed the charge to plus 40 millivolts, it has become depolarized. Now, this depolarization, what it's going to do is it's going to stimulate, it's going to cause the voltage-gated potassium ion channels to open. And so these potassium ions will diffuse down their concentration gradient into the tissue fluid outside of the axon. So now we will see this from that plus 40 millivolts, right, where we were depolarized, the voltage-gated potassium channels will open, potassium ions will leave, so the exoplasm will become more negative, right? It's losing positivity, and we can see that as it goes down. Now, we can see that there is this overshoot. This is called hyperpolarization because the voltage-gated potassium ion channels don't close exactly at the point of the initial membrane potential difference at negative 65 millivolts. They only close a little bit later, and that is why we have this overshoot, which is referred to as hyperpolarization. And then after that, the membrane will be repolarized, right? We will gain some more positivity, and then we will have the resting potential re-established. Now, what's going to happen is there will also be a refractory period. So this is where the voltage-gated sodium channels will be blocked. They can no longer open. And this is going to be called the absolute refractory period. So absolutely no electrical charge. So no matter the stimulus, it will not be able to charge any action. It will not be able to cause any action potential because the voltage-gated sodium channels are blocked. Then what will happen is that the relative refractory period will occur. So the voltage-gated sodium channels will be able to be opened. However, it will require a larger electrical charge than the initial value, right? It will have a higher threshold value. So a higher initial electrical charge coming into the axon membrane will be required in order for an action potential to occur and depolarization to occur. Now, the importance of this refractory period is so that the nerve impulse travels only in one direction. Okay, because if we were to have the action potential occurring, and in the region where it had just come from, the voltage-gated sodium ion channels were still open, then another action potential could be occur there, and so the nerve impulse would go in both directions. However, because in a region that has just had an action potential, there will be a refractory period, the action potential cannot be stimulated in that area, so it will have to only go in one direction, which is what we want. Now, this will also enable the maintenance of a specific frequency of impulses, right? Because there is this period, like in the absolute refractory period, where no impulse can be transmitted, it ensures that the impulses are going at a specific frequency. They're not going boom, 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 boom. They're going boom, 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 right? They're going in a specific frequency. So that's very important for the refractory period. So let's take a look at an example of how a nerve impulse could be transmitted along a myelinated neuron. So we have the myelinated neuron here, right, where the myelin sheath is acting as an electrical insulator, so no electrical impulse can be transmitted along that region. And we can see here we've had an action potential occur. So the inside of the axon is more positive compared to the outside, right? We have that plus 40 millivolt, right? We've had the depolarization occur. And over here we have a resting potential area where the outside area, the outside space is more positive compared to the inside of the axon, right? At negative 65 millivolts. Now, it's important to understand that the spaces between the myelinated neuron are called nodes of Ranvier, and this is where electrical impulses will be transmitted along the neuron. So in the action potential area, the inside of the axon, so the axon exoplasm, will be relatively more positive compared to the outside. And over here in the next node of Ranvier, the inside will be more negative compared to the outside, which is more positive. So because positive ions and negative ions, because ions of the same charge repel each other, and they also want to move towards a region of opposite charge. In this case, the positive ions want to move towards a region of negative charge. These positive ions that are within the exoplasm will actually move along the axon to the next node of Ranvier, where the exoplasm is more negative. Now, this change, the movement of these ions, will actually be what stimulates the initial voltage-gated sodium ion channels to open, allowing the generator potential to occur. Hopefully the threshold value will be reached for an action potential to then be stimulated. Now, as the positive ions move to a region of to a negative region within the axon, so do the positive ions outside of the axon. So the positive ions in that resting potential area will move to the more negative region at the action potential area. Okay, and so we can see that a local electrical circuit is being set up. Now, because the nerve impulses are transmitted from node of Ranvier to node of Ranvier, right? They're not being transmitted along each single part of the axon. It is called saltatory conduction because it's as if the nerve impulses are jumping from one point to another as they travel along the axon of the respective neuron. So that's really it for this video. If you found it helpful, take a look at some of the other content on my YouTube channel that might interest you. Otherwise, thank you very much for watching, and I hope you have a great day. Bye-bye.